Bacteriophages, teeny blobs of biological code that have the power to change the world. What if there was a weapon of mass destruction specifically targeted to just one type of bacteria? Some sort of biological weapon that would knock down a pathogen like listeria without hurting anything around it. Sounds powerful, right? Bacteriophages have that power. They are blobs of genetic code, DNA or RNA, surrounded by a protein coat or capsid. They're viruses that only attack bacteria, leaving other life forms alone. Well, okay, pedants, they also attack archaea, which are bacteria-like organisms as well. Their name, bacteriophage, means bacteria eater. They are tiny. As viruses, bacteriophages do the kinds of things other viruses do. They infect cells and cause havoc inside, taking over the host cell's replication machinery and making copies of themselves, eventually causing the cell to burst open and release the replicants. Typical virus stuff. They also do special phage stuff, like incorporating their own genetic material into the host cell so that it is replicated during normal bacterial reproduction. In this process, called the lysogenic cycle, the bacteriophage's genetic material either becomes incorporated into the bacteria's chromosome or forms its own circular replicon inside the cell. And by the way, certain mammal viruses can do this too, including the AIDS virus and the herpes simplex virus. Bacteriophages don't just insert themselves into other creatures' genes, they can also transfer genetic material between different strains and species of bacteria. That is, they transfer genetic material that is not theirs. The process is known as transduction, and it occurs when DNA from an infected bacterial cell gets packaged inside newly formed viral particles. This process plays an important role in the transfer of both antimicrobial resistance genes and virulence genes between and within bacterial species. Powering up bacteria against antibiotics. Antimicrobial resistance is considered one of the greatest challenges facing modern medicine and modern agriculture. When it comes to antimicrobial resistance, bacteriophages have a role to play as both friend and foe. As a foe, they are thought to contribute to the spread of antimicrobial resistance in bacterial communities. As a friend, they may prove to be powerful replacements for antibiotics in agriculture and human medicine. Antimicrobial resistant infections kill more than 4.95 million people each year, and they're in the top 10 global public health threats facing humanity. Researchers have observed the bacteriophage-induced transfer of antimicrobial resistance genes in both E. coli and Salmonella typhimurium cells in the lab and hypothesized that genes encoding for antimicrobial resistance in hospital effluent and in urban sewage could be transferred to bacteria in food supply chains, such as in fisheries, food-producing animals, and slaughterhouses. Virulence genes are also thought to be transferred by bacteriophages, including between strains of the important foodborne pathogen Staphylococcus aureus. As a friend, bacteriophages have been used to treat antimicrobial resistant infections in humans. Phage therapy, in which a cocktail of bacteriophages or a custom-made phage culture is administered to patients with serious and otherwise incurable antibiotic-resistant infections, and it's proven to be successful at treating persistent urinary tract infections, bone infections, and biofilm-affected implants. However, randomized clinical trials have not yet provided conclusive evidence to support its widespread use. Bacteriophages in food pathogen control. Dozens of studies have investigated the effectiveness of applying phages to food and food contact surfaces to reduce pathogens. Combinations included Salmonella on romaine lettuce, Salmonella on cantaloupe, Listeria monocytogenes on cut spinach, E. coli on alfalfa sprouts, and sugar toxin-producing E. coli on meats, with varying results. Listeria-specific phages have been commercialized as a pathogen control technology in recent years. For example, the makers of Phage Guard claim their product has an advantage over chemical sanitizers when it comes to destroying biofilms of Listeria monocytogenes. They claim it can be applied to food contact surfaces like conveyor belts during processing without imparting any off flavors to the food. The Phage control solution is purchased as a highly concentrated product that's diluted with dechlorinated water to 1 to 5% before use. The solution is sprayed onto food contact surfaces or persistent biofilms and can achieve 2 to 3 log reductions in a contact time of between 1 and 3 hours, according to the company's marketing material. At least one large food company is using the product to treat food contact surfaces in Australia, according to a representative of the company I spoke to at Australia's Food Pro last month. And I've got a picture in the post of a quite small bottle of Phage culture, which costs about $150. In short, bacteriophages are viruses that attack bacterial cells. They can persist in host cells without destroying them. This allows them to transfer their own genetic material and the genetic material of their hosts between cells. Such transduction can help antimicrobial resistance spread within or between bacterial species. It may also play a role in the transfer of virulence between bacterial pathogens. Bacteriophages have been used to control food pathogens on foods and food contact surfaces. The main source for this post is Colavecchio et al. 2017. Of course, there is a link to that in the post, and there's heaps of other sources, and they've been referenced inline using hyperlinks as well. So if you do want to check out the sources, check out the email or the post.